So I'll give you the sign off 2 minutes. You have half.
Yeah. Okay. No, we will speak. We will make a two voices speech. I will begin, and we will go back to chemometrics more than deep learning. In fact, we will just open a question on deep learning. We make a review on chemometrics about image fusion, about image and sensors. So, if I have well understood, yes, it was. So this is our outline. So, the original. So, first, when and why could it be needed to fuse image and spectrum, for example? So, for online monitoring, here you have the example. It's from Basil processing to like pesto, I think. And here, for example, you have a vision system. No.
No, you have to do it virtually, just without touching.
Okay. So for process online monitoring, for example, but also for quality control. Also in agriculture, precision agriculture, for example, there are a lot of imaging from drone or machine, embedded in machine, and also spectra measurement or other sensors that can be fused in order to characterize zones in the crops, etc. But in fact, actually, it's yet not so many applications, and most of the applications are based on RGB image plus other sensors. Okay. So some words about image structure and issues. When you have hyperspectral images, you can see this as a cube. It is always said as hypercube, but in fact it's a cube. And in this cube, you have monochromatic planes. That means that you have a lot of images at single wavelengths. Okay, you can see that. Or you can see for each pixel, you have a spectrum. You have these 2 points of view. And a degradation of this hypercube is multispectral. For multispectral, we have only combination of bands like that. I think that we have only a small number, but the limit between hyper and multispectral is a bit fuzzy. Okay. So you have different resolution, and you have to handle spatial and spectral information. So 2 perspectives of analysis from spectral image, hyper or multispectral image. You can either look at the spectra. Okay, so that means that you unfold your image as a matrix, and with this matrix you can do classical chemometrics, or you can watch to this cube like a lot of images and process that with image processing. So, for example, here you can make spectral analysis, and but if you do that, you completely lose the spatial correlations. That means that all the spectra you have here are completely independent. Okay? And if you do the reverse, that means that if you, for example, you make some texture analysis or region or segmentation, you can do that on each wavelength, but there is no spectral correlation. Okay? It's a reverse. So, classical. So, if we focus on spectra, so we do pixel-wise unfolding. You can do multivariate analysis, exploratory, like PCA, classification, calibration, etc., etc. Here you have example with a PCA, and what is very interesting is that if you unfold and you make a PCA or MCR, etc., when you have scores, you can refold and produce images. So it's very interesting because you have images of concentration, or images of load, of scores, and you have a relationship between the loadings and these images, so you can do an interpretation, but a posteriori. Okay? And then, so I think it's your turn.
Okay, so this is the spatial side. If you look at each image, you take a specific spectral wavelength and you look at the image. Then there are several ways in which you can analyze the spatial relationships. One of the most popular is to take so-called textural analysis, so textural index, for instance, gray level concurrency matrix, but sometimes even simple things like to take just the average of the power, of the energy, whatever you want, or the entropy, or the local entropy of the images. And other things you can do, you can calculate the distribution of the pixel intensity at that specific wavelength, and you can treat this distribution as a signal because you can fit the shape, and so you can represent actually an hypercube with a long signal of histogram. In all this way of treating the hyperspectral image, what you lose is the direct correlation with the spectrum. Which are the wavelengths that are responsible for specific pattern. So this is a way to extract features, but you are more focused on the spatial relation than the spectrum. And then there have been several proposals how to take into account both the spatial and the spectral relationships. And this in chemometrics has been done in different way. And the proposal, I would say, any day are increasing. So this was one of the first multivariate image analysis, the work by Baratti and McGregor. So the idea was simple. We unfolded the image, but then we add also the unfolding of the neighbor pixel. And we can do this for all the spectral channel. In this way, when we do multivariate data analysis on this unfolding, we will take into account of the spatial correlation because we have the neighbor pixel in the column. And we take also into account the spatial, so we can have both things. And this is a different way. This was a proposal by our research group. So instead of using augmentation with neighbor pixel, we can use filter. So this is very close to the convolutional layer. But in this case, of course, the filter coefficient are fixed because we select the wavelet transform, which is one particular filter. And so in this case, what we do, we filter a different level, a different scale. So we do also, let's say, data augmentation because we start with one image and we have several because we have the filtering of these original images. But what we keep from the chemometric side is that at this point, we analyze this data by standard multivariate tool, PCA, PLS, MCR, whatever you want. And this was also a proposal that is more for exploratory analysis, but can be used further. So this was a bit more dedicated to hyperspectral images because the previous approach was more for multispectral. So in this case, we really do, in the first part, a bit of what you do with the net, not knowing it. Because we do exploitation, so we use a filter, we use wavelet decomposition again, then we do compression of this information, we call this encoding, but actually it's a compression by extracting feature, by gray level concurrency metrics. Then we use PCA on this compressed data to select only which are the most important descriptors, which are the most important feature. So we use in this some concept from essential information. So in the PC space, we take only the extreme, and taking the extreme means that of all this exploitation, we select a much smaller subset of this image. So at this point, we have a data cube, but it's smaller than this one. And we can call this the informative data cube, because we retain which is the most informative of all this huge data. And we can unfold this and do multivariate data analysis. And what is nice with this is that we can have a calibration image. So we can do some multivariate data analysis on this. From this, we can recover special partners, but we can also recover which are the important spectral wavelengths that are related to this special partner. And if the images are not so different from the original one, we can just do projection of the new image in this very last PCA space. So we only need the filtering step and then the projection. So it's much more fast than building the model. And we can recover the partner in the new image. So this is somehow was something that we did with standard chemometrics tool. Then, for managing spatial and spectral information, there is also several other proposals, and one of these is to use tensor algebra and tensor model. So we can actually see the image in the cube, and we can decompose the cube directly, taking into account both spatial and spectral dimension by using tensor decomposition. And one method which is becoming popular is the so-called block term decomposition with the LL1 rank. What does it mean? It means that we do a decomposition like in the picture. It's like a parafact model for the one of you that know parafact or can decomposition, CP, how it's also called it, but with a constraint that the third mode, which is the spectral mode, has rank 1 in each of the factors in the decomposition, while the spatial mode may have a different rank from one factor to another, or could be the same, it depends, but this can be more than one sub-factor. And both sub-factors should be the same so that we can reconstruct the spatial dimension. And this can solve the problem that I may have a complex spatial structure with the same spectral signature. If I do a standard parafact, in this case, I will have several components with the same spectral image. And so I will have to fuse then all these components in one to see the spatial scene similar to the ground truth. In this way, seems more easily to recover the ground truth. And there is also someone who has combined this approach with convolutional network somehow in the literature. But this is something interesting also. And
so I will present another method, starting from the constitution to constituents that, in fact, we have two separate communities. We have for processing images and for processing spectra, we have completely different tools and paradigms and all different. So we have chosen to separate. So if we start from a hyperspectral image, we can have some region, or we can show some images with a central pixel. Okay, this is this sub-images. So each of these sub-images will be analyzed separately for the spatial and the spectral dimension. Okay, for each of this image, we can project each of these image on PCA component or just taking wavelengths as you want, but we can generate monochromatic images. Okay, so in this part we have only spatial structure, but we can make multiple point of view with several PCA components, for example. And on each of these series of images, we can make image analysis, what we want. Okay, textual, segmentation, what you want. But the result of these images are, for each of the images and images, we have some indexes for texture, etc. Okay, so we have here spatial information, which has been processed by dedicated spatial processing. And here is the reverse, it's the opposite. We unfold each of the sub-images, so we have matrices of different lengths, but the same spectral dimension. And on each of these matrix, we are doing chemometrics. For example, we can just take the mean spectrum or do an MCR decomposition or what we want, or SVD, and we retain the first loadings. Okay, but the aim is to obtain here and by lambda, by a certain number, spectral information. In these matrices, we have spectrum. In these matrices, we have only textual indexes or what we want, or number of regions, etc., or morphological. And these two set of blocks are then fused to produce new scores. Okay. And here, it's a general framework. You can put what you want as tools for the spatial, for the spectral, and also for the data fusion. Okay. We have developed that in the framework of the thesis. And the application, but we skip it because that time.
And with this slide, what we try to figure out is what we may need to fuse, coming back to the first slide of the presentation. So we may need to fuse images that comes from different technique, and we will see something afterwards. And if we have to fuse images with spectral sensor or other type of sensor, then we have to translate the images in a dimension which is monodimensional to be compatible with the single sample on which we acquire the spectra of the or other sensor variable, no? Because the sample is one, so in the image we have a cube, but in the other sensor we have just a vector. So there are several ways to obtain 1D features from the image. So for instance, one of the most popular is to select a region of interest, and so just to take the average spectrum in this spatial. So you unfold the pixel in a region and you take the average spectrum, so you have a 1D vector. Of course, you can do textural, you can do textural features at each wavelength, and in this case, you also have a 1D vector for a single sample. You can do a mixing, for instance, with MCR, and then you can have a textural feature on the concentration map. Instead, for each spectral wavelength, for each component that you have resolved. And this component, you can store the spectra, so you can come back when you need to do interpretation. You can use the approach I showed you before, so we can extract feature, or you can use the spectrogram at each wavelength. So also in this case, you will have a collection of spectral histogram, and this is also has been proposed. What I have put there is the two dimensions, spatial and spectral, if you have a direct or an indirect linkage with that. So for instance, in the first case, you lose completely the spatial because you do average. In the other case, you recover the spatial either directly or indirectly, and the same is with the spectral dimension, because of course, if you do a mixing, you have in memory a pure spectra that correspond to one component. If you do this stacking of lambda, then you have the correlation among the different lambda that is somehow on top of the correlation among the different features. So then you need some post-preprocessing if you want to highlight one thing or another. So that's what I mean with a small table in the end. Okay, if we speak about fusing image, of course we can use approaches like the one suggested by the Barcelona and Lille group, fusing different platform by using multiset. So You can have a different image. This can be taken by different platforms. Of course, in this case, you have one problem because you have to make them of the same size with respect to the spectral dimension, not the pixel, but the spectral dimension. And then you can unmix and then you can recover the difference. And this is a proposal. If you want to do punch sharpening, then Jean-Michel will show other methods. But within the context of multivariate curve resolution, you can do this if you first manage to make the images of the same size, maybe with binning, and then you treat this as an incomplete multiset. And this is an example with Raman and fluorescence. And of course, you have some assumption. The assumption is that pure constituent should be shared between the two images. That the chemistry behind should be similar, at least for some component, not all, but for some. Also, you can use coupling of tensor and tensor factorization. So here you have a general loss function. If you want to do joint analysis of two tensors, the same applies if you want to do joint analysis of a tensor and a matrix. If you assume that one of the mode is in common or is not in common as in this case, but related, so you can have a term in your loss function that say that more or less, for instance, the spectral dimension in the decomposition should be equal or should be similar up to a small function, then you can solve this and you can recover all the factor matrix that allow you to combine together the images. And also this concept has been applied to punch sharpening, because in this case you have a high resolution image that is the one you want to estimate, and you have a low spatial resolution image and a low spectral but high spatial resolution, and so you can decompose these two all together and to recover what is missing once you have recovered the factor matrices. So again, we have this different perspective to fuse image all together.
Yeah. I will add some more about punch sharpening because we already spoke about that. And punch sharpening is an idea inherited from remote sensing. In remote sensing, they are doing that. They have been doing that for many years. Okay. So it's in fact it involves merging high resolution spatial, low resolution spectral with high resolution spectral but with low resolution spatial. It's a bit like if you want to colorize black and white images. You have black and white images and you want to put some color on it, okay? And if you have no indication, you can put a red, black, blue and green, whatever you want in this image. But if I told you this is blue, this is red, maybe you have some indication and you can spread the color on the image. It's the same here. We have spectral images, so we have high spectral resolution, but very low spatial resolution, okay? And here we have some pan-chromatic images, I mean black and white, okay, with high spatial resolution, very high resolution, okay? And the aim is to merge these two in order to produce high resolution in both spatial and spectral, okay? And it's very common in remote sensing. And I will tell you two algorithms because there is two main historical categories, either component substitution or multi-resolution analysis. So the first one is component substitution. So we start with a multispectral image, so we have a lot of wavelengths, but very, very low spatial resolution. We have only some points, let's say, okay? And here we project that on a latent space, for example, doing a PCA. That means that here we have only some scores, images, with low resolution. And then we interpolate these images, monochromatic image, to the resolution of the pan sharpening, which is here. Punch to the pan image, to the pan chromatic image. Okay, so you are just doing square, little square, but with the same, in fact, resolution. And then we compare the panchromatic image with each of the plane, for example, thanks to histograms of gray level, and we select the one which is the more resembling this plane image. And we substitute this with the panchromatic image, and we go back in the reverse. For example, if we have done here PCA, we do the reverse PCA. And doing that, we produce something which is an hyperspectral image. It's a false vertical image, but it is done like that. Okay, 5 minutes. Okay. Oh. The other way of doing is multi-resolution analysis. So it's not exactly the same. It's a reverse, in fact. From multispectral image with a lot of wavelengths, we make a spatial interpolation in order to have the same spatial resolution as the panchromatic image. So we have a lot of planes, okay, with spatial, but you can see here we have the same pattern, in fact, but with high resolution. And we compare each of these planes with the panchromatic, and we compute gains, in fact. These gains are based on the resemblance between the planes and the panchromatic image. And we apply these gains, and we go back to a perspective image. So you can see it's complementary. So this is a tool. And we have applied that for phenotyping, to phenotyping plants. So we have a student, some of you can recognize Maxime, who has measured 2 spectra of the vegetation in mice of cones, and I've done chemometrics, classical chemometrics with that, because we had an experimental design, irrigation, without irrigation, with genotypes, etc. So we have, thanks to ASCA, we have extracted loadings, okay? Loading representing, for example, the robustness of the genotype regarding the drone, okay? And then we have used a drone with RGB image, simple RGB, and some spectra. That means that this drone has embedded a camera and a spectrometer. We have done pan sharpening on that, so we have reconstructed a false hyperspectral image, but we have applied directly the loading on this, and we have obtained score maps, in fact, which are a very, very good which were very, very useful for the growing, for interpreting the genotype and environment interaction. So this is an example. Another example is fusing different hyperspectral images with different modes and different resolution. Here we have infrared, fluorescence, and Raman. And these images are from different modes, so different spectra, but also have different resolution. So the problem is to fuse images with different resolution. And the trick the authors have used is they have taken the most resolute, the less resolute image, it was infrared image, and for align the same resolution for the other images, and that means that they have small images, and they have unfolded these images in a third dimension. In this, doing that, they have common dimension here, and they were able to apply trilinear multiple coinertial analysis, and at the end, they have obtained score images for each of the modes in order to represent here it was xylem, phloem, etc. So it's time for conclude.
Yeah.
For conclusion.
We go fast here because we have seen
Yes.
many things of CNN, so you don't need this. I just remind that there are many parameters to tune. So this is something that as a chemometrician, I see as a huge problem in applying this method. And this is an example from the literature of people that want to recognize fat and meat in lamb, and they apply a convolutional neural network, but they do different things in the 2 dimensions. What is relevant for the discussion is that within the image, they take a soup cube, so only 6 spectral channels, and they apply 3D convolution. So this means that they use a filter that mix spatial information with spectra. And this to me is a bit odd, because I don't know if there is really sense to some, I mean, intensity of two close pixels and to mix this with a different spectral band. So that's why I show this to you, because we have several questions. The first one is, which are the scenarios in which we need to go deep? And we had some flavor this morning, but I think we can discuss it more. How to do data augmentation with hyperspectral images that have these two dimensions. Does it make sense to mix? Oh, sorry. Dario given us.
You're creeping in.
Yes.
Okay. So this was my previous question, and also this, because if you do data augmentation in a standard way, by crop, rotating, translating, does it make sense with an hyperspectral images? I don't know. And one other thing is that as chemometrician, we are very keen and willing to maintain interpretability, both in the spatial and spectral domain, and especially which is the link between the two. So that's something that I don't know how far can be recovered if we go in a deep scenario. So my personal idea is that it seems to me that when we have a sort of huge database of spectra, like a bit what you show, Martin, in this scenario, we have a sort of quite a complete vocabulary of the chemistry that can go in a spectra for a given property. So using methods that rely on complete knowledge of the vocabulary, I think could make sense. But if you acquire new data and you have your data set, your limited one, I don't know how much it is worth. And I would like to understand from people that is more expert than me in using deep learning, a better this concept of fine-tuning a model that was developed in other example. Because maybe this is the clue, but another thing is that I'm convinced that we have a lot of chemometrics method that actually do this exploitation, compression, and information recovery, but simply we don't call it with this name. So there is the need of doing this translation and link between the two communities, because I'm really convinced that we already have many tools and we do this, but we are naive, so we don't say that we do this. We call with other names.
Okay, thanks to speakers. We have time for a couple of questions. I just want to make an introduction comment here, is that I think there's a lot of space to work here. Usually, chemometricians are very crafty people. They pick up numerical methods from different disciplines and they adapt it to the problem and they concatenate things. One of the advantages that I see in using neural nets from other systems is that you can do end-to-end. You can just input your image and then you can train it to do something. What you lose is control over every process and probably the explainability.
Yes.
So I know that for the chemometricians that is very important, of course, because you want to learn about the chemistry. Okay, so that is an aspect that has to be incorporated, and that's why we shouldn't let people from computer science just do this kind of models to find an answer, but as you were saying, interact between chemometrician chemists and physicists in order for the model to be able to say, okay, I'm choosing that wavelength because this or that, okay? So with that further discussion will be a nice topic. So a couple of questions, Hakim.
Just and a comment to your third question. So the infrared imaging, it makes a lot of sense, and I can give you an example. So, the infrared microspectroscopy, that's what I guess what you're talking about when you say infrared imaging, you have in the spectral domain a lot of information about morphology, simply because of the sizes of the object cells. They give you scattering, and the scattering gives you really nice information about morphology, refractive index, etc. And the same type of information you have in the image plane. The image plane also gives you the morphology as a so depending on the chemistry, etc. So this way, the correlation, or there is already a correlation, you can say, between the image and the spectral plane. If you look at these quantum cascade lasers, which have a high coherence in principle, which are used also for imaging systems, they also give you a lot of scattering phenomena in the imaging plane in principle. So you also have the other way around. So it makes a lot of sense to look at this correlation in imaging.
But because the objects are in the same order of
Yes,
magnitude
but it's
side like the wavelength.
That's what it is.
Yeah.
This was an example of
Yeah.
infrared imaging, so it makes a lot of sense.
Yes, one more question before Harold. Yes, Tom?
You a little bit confused me with the 3D convolution, but so I will ask another question. So your fusion kind of with the spectral and the spatial part goes into some, then spits out the parameters, which is kind of like trying to compress the hyperspectral cube. And you are trying different bases to do that. So is there a way to kind of find the right basis? Because yeah, it would be cool to find the best compressible kind of representation of your hyperspectral cube, which result then in the second parameters which you are after.
I'm not sure I got the.
Yes, I think that this is a stake of analyzing spatial and spectral simultaneously and to find this basis of projection directly. I mean, this is your question.
It's just you seem to treat who want to separate the problem.
No, I mean, we have shown that there are several approaches. And in some approaches, you separate and then you try to find the relationships by mixing the feature that you have extracted distinctly. In other approaches, you go directly to take into account them both, like with the tensor algebra. And in other situations, you stay a bit in between, because you try to have a data matrix that is augmented with the spatial information, and then you analyze this together with multivariate modeling. Because when you do modeling, then you benefit from the correlation structure that you have in your column. So if your column contains both information, you will take this into account.
If I rephrase, if I find the best basis to compress the hyperspectral cube, is that the goal? And do I learn something then about the system? Is that what you are trying to do?
Yeah.
Finding the best basis?
Yeah, basically we are seeking a latent space that can represent this relationship.
Yes, exactly.
And that is interpreted.
Yeah.
And it should be interpretable.
But then that kind of doesn't go against, that's how you confuse me, because that doesn't go away, it doesn't go against the convolution part.
No, it's, I don't see it against.
No, no, it's not against, it's
It's not
another
against.
way of thinking.
It's a different perspective, I would say.
Okay, let's thank the speakers again.